registered it?
Well, I don't know how they should have written the code down. But that doesn't mean they did. We should know what his code is. We could actually go look up those codes.
Okay, so should I just keep those?
Yeah, just hang on to them. And what's that one?
I don't know.
You don't know what that one is? Oh, it is one. Okay, so here, put that away. I don't really need this. Okay, good morning. Let's get started. Everybody go trick-or-treating last night? My wife won't let me deliver candy at the door because one year I dressed up as Belnar, the conehead. I gave away fried eggs, 6 packs of beer, and cigarettes, and she wouldn't let me do it. But that was actually a skit Saturday night. Fried chicken embryo, packs of 24 rolled paper with stuff with carcinogens that are flammable, and meat. So, I hope you did see the announcement about that seminar with the medical school. It is exercise 4. Michael Dean is a renowned cancer biologist, and so he'll be talking about, he heads up the human genetics section at the NIH, or National Cancer Institute, and so I think his talk will be pretty good for the interested in cancer. What we need to do today is, let's look at the learning objectives and see how far we've gotten. So, contrast the different types of coding mechanisms as to similarities and differences. Well, we can't do that because we've only looked at one, right? So we've done class and coding. We're going to do another type of coding in detail, and those are the only two, so that you'll be able to compare similarities and differences of the types of coding. and then discuss how vesicle is targeted to a specific membrane destination. The concept that you should be developing is almost the same as turning on a gene. What happens is you've got some of the same players, but it's a unique combination of all of those things. How the lipids are phosphorylated, which coding mechanism is used, which targeting and fusion proteins. And so we don't even have all the players, but we're starting to assemble a bunch of things, and it's a unique combination for every vesicle that has a specific destination. So we're trying to build that concept up. And then that's going to allow us to make some useful predictions about what happens when things go wrong, and actually the basis for certain medical treatments. And eventually we'll get to the next structure. So we've all sort of been focused on the ER so far, but we're going to get into the Golgi because that's where we're driving traffic to. So to get there, we have to take vesicles and bud them off the ER and fuse them with the cis-Golgi network. That's the next compartment. Okay, to do that, we use a unique coding called COP2. So that's going to be our poster child for that type of coding. And it is similar, but it's also got some differences. So what we're doing is this one right here. We're going from the ER, budding off this vesicle, then it fuses with the cis-Golgi network. Remember, the ones on either end of the Golgi stacks are always bubbly looking because there's so many, they're either fusion or budding events that are happening at the sort of the indoor and outdoor of this Golgi apparatus. So this is the type of coding, COP2, that we're using. And this is anterograde, okay? So to do that, we use a small GTPase, surprise, like the one that we used previously for Clathren. We used ARP, right? So this is in the same family as ARP. It just is called something slightly different. And so here's the setup. Again, this should be very similar to what we discussed last time. When it's bound to GDP, it's soluble. And so it's got its landing gear retracted. Okay? And what's going to happen is, so it's got this amphipathic helix. That's just a biochemistry speak for a type of helix that is embedded in the plasma membrane, and it's also got a little extra hitch to it. It's actually, it's not shown here, but it's actually got a rubber tail that extends out of that, just like RAS has, and so that's going to help it stick into the membrane. What does it need to do to be able to do that? It's got to pick up GTP and get rid of the GDP. How do you get a GTPase to do that? Well, if they're monomeric like this, you need a helper, and actually for most GTPases. So it's got to run into its GEF. And this is the SAR GEF, and we know what that protein's name is. It's Sec12. So that means it was identified in yeast as secretory mutant number 12. And so it's a transmembrane protein. It bumps into that. So it dumps the GDP, it picks up GTP, and then it lowers its landing gear. And so that allows it to stick to the cytoplasmic leaflet of the ER. Okay? Now, when I was drawing that for the Clathren coding, I think some people got the idea that the ARF was on one side of the membrane and the coding mechanism was on the other. That's not true. The ARF, and in this case SAR, has to be on the same cytoplasmic side as the coding mechanism, because the coding proteins are coming from the cytosol. Okay? So let's keep that straight. So this is in the cytosolic leaflet. And so what does it do? Well, let's think about what did ARF do? It recruited assembly particles, correct? So the analogous situation here is a couple of proteins. This is a heterodimer. It's called Sec2324. And the Sec24 subunit actually recognizes the cargo receptor, just like the assembly particle did, right? So it recognizes the cargo receptor, and it needs to have that cargo receptor because the cargo is a soluble ER protein that needs to go to the Golgi. And the problem is the coding mechanism can't see inside the ER, but it can see the cytoplasmic domain of a transmembrane protein. So that's why the cargo receptors are always transmembrane proteins. So they display some sort of sequence to the coding mechanism, say, here I am, okay? So this is going to coalesce onto the ER because it's recruited by SAR. So the role of the GTPase is to recruit the coding mechanism, just like it was for Clathrin. So Sec24 binds to this. Sec23 is actually the gap for SAR, okay? Except it's turned off right now. It has to be turned on in order to do its job. What does a gap do? Now that we know the GTPase cycle. You know what a GEF does, right? We've already talked about that. That causes that to happen. What's the gap supposed to do? Right, it causes the GDP to lose a phosphate and return to the GDP state. And so that gap stands for GTPase activating protein. Where's the GTPase? SAR is a GTPase. It just has no ability to turn itself on. Okay? And when it does, it's going to turn itself off by splitting the terminal, the gamma phosphate off of GTP. Okay, so, but we need some other players. We need to create this lattice, just like this is not quite the same soccer ball. It's a very different shape. But this lattice has an inner coat and an outer coat. In this case, the inner coat is the Sec23-24 SAR complex, which then recruits the outer coat, which is Sec1331. Okay? And so the outer coat, once it comes in, it completes the lattice and deforms the ER membrane into a vesicle and causes budding. Okay? So it's the completion of the lattice, the inner coat and the outer coat, that's going to pull it off into a bud. Now, we're not going to worry about what chops it off. There's got to be some protein that does that. So let's say it buds off. What's the first thing that has to happen? You should know this by rote. Well, it's got to pinch off. As soon as the vesicles form, that's true. You got the cargo. It has to what? It has to uncoat. And why is that? You ought to just know this straight away now. Yeah, it's got to display a targeting and fusion complex that we're going to talk about today, right? So we have to uncoat it. So the way this uncoats is Sec1331 can detect the deformation in this curvature, okay, which activates it. What is its job? It is a gap out. It's a GTPase activating protein, a gap out. I mean, I just made that term up, but what it is it's activating 2324. Okay? So the gap needs to be kicked in the butt, turn itself on. What does the gap do? It turns on SAR's ability to clip off the terminal gamma phosphate. So you got to have three things. You got to have Sec31 has two roles, 1331. It completes the lattice, causes budding, the thing pinches off, and then at a certain conformation, the bending of that protein activates it so that it talks to Sec23. It says, get your gap activity going. And it turns on 2324, so it becomes a GTPase activating protein, and it talks to SAR and says, you need to split your terminal phosphate, and that's what it does. So it takes three of these things talking to one another to uncoat. So once you split, so why does that uncoat it? Well, it's because when SAR is bound to GDP, it retracts its landing gear. And so now you've loosened this whole lattice, and so it unpeels and comes apart. And then that displays various proteins that we're going to talk about that will help target it to the destination, which in this case is the cis-Golgi network. That's the target membrane. This is the donor membrane. The target membrane is the cis-Golgi network. Okay, so did I lose anybody? Anybody have questions for what's going on here? So it's a lot of similarities between this and the Clathrin coding business. All right. Now we need to talk about that targeting and fusion complex, and to do that, we need to bring in another set of proteins, and the first one are the RABs, okay? And I guess let's go ahead and do these problems before we get into the RABs. It's a good break. So get out your clickers. Get my polling device up here. Get this out of the way. It's going to be hard to. Okay. Okay, polling's open. Try to do this on your own. The way to do this is to draw the protein as a linear molecule, and looking at that, be able to predict how it's going to fit into the membrane, if it does at all. See how many people. Stupid thing. I'm sorry. Oh. I'm sorry, I didn't check that. Well, okay, well, let's tell you what we'll do. Is it not letting you put it in without, okay. Let's start over. All right, polling closed. Okay. All right. 5. Continue. All right. Polling open. So it's open. All right. See if you can answer it now. Details. Continue. What's wrong with it? All right. I guess that's not going to work. So I don't know what the problem is. Okay. So now let's go ahead and you've had a chance to do so. Now turn to your group and come up with a group answer. I'm going to randomly pick a group. Does anybody have a consensus? You guys want to try it? What's your pardon me? D. Did you say D? E. Oh, E. Okay. Does anybody have a different? So E, N-terminus will be cytoplasmic. We'll have one transmembrane alpha helix. Anybody have a different? Okay, good. We'll just move on to the next. So the key thing is to figure out how many alpha helices it has and understand the orientation. So the minute you see that this has an internal start transfer, you know that there's no signal peptide, so that N-terminus is not going to necessarily be luminal. That means it could be either or. And you have to look at the charges adjacent to the start transfer sequence. And since it's only got one alpha helix, it's only going to have one transmembrane sequence. And it says that the positively charged residues border the N-terminal side, and the rule is the positive side's directed to the cytoplasmic compartment, and then the negative side will put it in the lumen of the ER. So it is E, in fact. It's N-terminus will be cytoplasmic, and it will contain one transmembrane alpha helix. Okay, good. So I think we have one or two more of these. Okay, let's do this. This is more vocabulary question. Try to do this by yourself. Okay, then turn to your group. Get a group answer. Okay, randomly, group 7, lucky 7, where are you? Unlucky 7. Where are you? Okay. So, what's your answer? It's not F. What? C. Okay. Anybody have an assembly particle? Anybody have a different answer that you'd like to defend? Oh, good. It's another consensus. All right. So that is true. It is and if this is clathrin at the plasma membrane forming a coated pit, then it's got to be a particular assembly particle, AP2. Okay. And AP assembly particles the particles are composed of adaptants. And which adaptant is it that binds the cargo? Mu. Okay. It's mu. Right. Good. So let's see. Maybe one more quick one. So this is a little tricky, but so you have an ER resident soluble enzyme. And what would be a good example of an ER soluble, not a membrane spanning protein, it's no alpha helix that spans the membrane, so it's soluble floating around. What's a good example of an enzyme that's ER, does its job in the ER? So if it accidentally escapes to where? What's the next part? To the Golgi and what's cisterna? Cis Golgi network. If it gets there, it needs to be sent back. Okay, so this is retrograde transport from the Golgi back to the ER. What mediates? What coding mechanism mediates that? There's only 3 choices. And you don't know what this is yet, theoretically, so that's not the answer. It's COP1. Okay, so it is COP1, and that's a little confusing because COP1 is going in reverse. COP2 is actually the first step, but they're named in the order they were discovered, not in the order that they are in the secretory pathway. So I think I have an explanatory. Yeah. If we go to look at the key, and the key is up here somewhere. Is this it? Yeah. There you go. So COP1 is for retrograde back to the ER and between the Golgi cisternae. Okay. And also right there, this is constitutive transport to the plasma membrane. Okay, so those are the COP1's used quite a bit. Okay. Good. So let's go back. Now, what we want to do is we've got this targeting and fusion complex. We need to understand what composes it and how does it do its job. Right? So that's the next step. And to do that, to develop it, we need a couple of new proteins. One of the big ones is the RAB GTPase family, and it's kind of like ARF and it's kind of like SAR, but it has a very different role, okay, because it's used in targeting and fusion. The reason it's used in targeting is there's a specific RAB type for each type of compartment where the vesicle is headed. Okay, so if you look at this, here's the key. There's almost a different RAB for each place you want to go in the cell. Okay, and I'll direct your attention to 3 different examples that we'll look at very briefly. We'll spend our big example, our poster child of RAB is actually RAB1, and that would be to direct a vesicle from the ER to the cis Golgi network. So it would be taken up in that coding process. It would be recruited into that vesicle. So the RAB is going to ride with it. Okay, and how does it get there? How does it get attached to that vesicle? Well, and so that's RAB1. So we'll look at that story first. This one is important because it's important in insulin signaling. So it's a part of secretory granules that fuse with the plasma membrane. So that's why we'll look at RAB3A. And RAB5A is shown in some detail, and that's the one that's on the plasma membrane and is taken into clathrin-coated vesicles coming from the plasma membrane, fusing with the early endosome. So we'll look at those in a little bit of detail. The rest of them, you don't need to memorize it. What you do need to understand is you need a different RAB to go to different places. Okay, and so a RAB functions just like our other friends. Okay, when it is bound to GDP, it deploys its landing gear and sticks to the vesicle. So it has to bump into its GEF to do that. But it's a little bit more complicated than that because, let's see if I can get this to go. Yeah. It has another protein that's controlling its activity. And so here's a Joe Blow GTPase cycle. And so you're familiar with this stuff right here where RAS is an old friend of ours, but so pretend this is RAB, okay, and it has to bump into its GEF, and it's gonna lose its GDP and pick up GTP. So the GTP comes on, and that drives it into the on state, and so that it lowers its landing gear and sticks to the membrane. And then it has to encounter its gap, and then that's going to turn on the GTPase activity intrinsic to RAB, and it'll split GTP and return to this state. But what happens here is it has another protein. It's called a GDI. guanine nucleotide dissociation inhibitor. So this is so important, it's got an extra level of control. After this thing dumps the gamma phosphate and returns to the GDP state, it recruits a cytosolic protein called GDI, and that, what it does is it protects this GDP from coming off. It prevents the dissociation of GDP, okay? And so now the GEF is going to have two roles. When RAB bumps into the GEF, it first has to dump the GDI, and then it can exchange the GDP to pick up GTP. So it just has a different role. But understand that RAB, like ARF, like SAR, they are cytosolic proteins floating around until they pick up GTP and deploy their landing gear. Okay? Where does this one go? Well, this one sticks to, typically it sticks to the donor membrane and is recruited into the vesicle as part of the targeting and fusion complex. Okay, so why is RAB so important? That's the next question. Okay. And the reason it's important, let's turn this off, is because RAB is going to recruit what are called RAB effectors. That is, they do the work of RAB. RAB recruits them, and then they do the work of targeting. Okay? One of those RAB effectors is called a tether. Okay, and so this is an example of a tether. And typically, these are long, single-pass membrane proteins that extend outwards from the target membrane, but they usually are extended from the donor membrane as well. Let me give you a specific example. So this is, I'm sure most of you have played tetherball, you know, that pole, you go to camp and you hit people in the face with it, you know, it swings around and you try to duck it. So, but it's a long tether, right? The ball is tethered to a pole. And so what this is a crowded place and there's stuff flying all over the place. And so you try to grab on to the vesicle using very specific snares that are flailing around. So this vesicle may be bound to a motor protein zipping past, but the tether will reach out and try to grab it. And so let's look at a specific example of a vesicle that's coming off the ER. Okay. So we have the ER, here it is. So that's the donor, right? And so it's just, you just butted off this vesicle, you dump the coating, and now you have RAB, but you don't have just any RAB, you have RAB1, because RAB1 is tasked with the important function of moving vesicles from the ER to the cis-Golgi network. Okay, so it's very specific. And so this RAB is going to recruit a RAB effector, which means it's a helper, right? So it's a protein that does the bidding of RAB1, and this RAB effector has a very specific name. It's called protein 115. This was obviously made by biochemists that had no creativity and just named it after its molecular weight, right? So this thing is flailing around, and it goes to the bubbly cis-Golgi network. Okay, and guess what? The cis-Golgi network has its own tethers. Now this is a more inventive name. These are called Golgians. This is obviously named by a biologist. And so these tethers wrap on each other. So this is a long extension of bringing you into the target membrane. So that's the role of the tether. And these are called RAB effectors. Yes. Well, tethers in general are a class of RAB effectors. So they do the bidding of RAB. RAB recruits them, they do the work of snaring. I don't want to use that word, but that's not a protein, but they tether. Now you've got a putative vesicle ready to dock with the target membrane. So at that point, you're going to need another set of RAB effectors to go forward. Okay? And a better example, so there's going to, in a minute, we're going to use these guys for something specific, but we really don't consider them RAB effectors. What are other RAB effectors? Okay, so what happens is, and now we're to a different membrane, so ignore the fact that this is now RAB5. Where's RAB5? RAB5 is targeting vesicles from the plasma membrane that are encased in clathrin, and they lose their coating, they're headed to the endosome. But that's where RAB5 does its work. So I like this picture because it shows the whole cycle. So it gets converted to the landing gear site, it digs into the membrane, it becomes active after it loses its GDP and picks up GTP. At that point, it recruits its effectors, which are tethering proteins, filamentous tethering proteins, and this is a kinase, and this is a lipid kinase. Remember that part of the information to tell a membrane vesicle to go to a target vesicle is the type of phosphatidylinositol phosphorylation pattern it has. And of course, you have to have a kinase to phosphorylate the lipid head group. Okay? And so PI3 kinase is going to put a phosphate group on the third position of the phosphatidylinositol ring. Remember, it's a 6-membered ring. I think we can bring that up. Let's just remind ourselves what that looks like. Yeah, here we go. Okay, so it's a 6-membered ring. This is 1, 2. It puts a phosphate group right there, and that's going to recruit a class of proteins based on the shape of that negative charge on the lipid membrane. It's going to tag that membrane as this is your destination. Okay, so that's what we're going for. Information about where to fuse. Okay, so that's a good example. Lipid kinases are a good example of RAB effectors that tell it where to go. Okay, and then there are a host of other proteins that are involved here. These green things could very well be motor proteins. Good example of the third type of RAB effector is a motor protein. Why is that a good idea? Well, it's because this is not random at all. There are a series of highways that run from the ER to the cis-Golgi network. They're called microtubules. And so this vesicle can hitch a ride with a motor protein if it's recruited by the RAB protein. So the RAB will recruit something called kinesin that we'll talk about later. It's that thing that walked in the video carrying the vesicle. It recruits that motor protein, and it travels along the microtubule until it hits what? It gets close enough for the tethers to wrap around each other. And so then it's done its job. So motor proteins are a third type of RAB effectors. Okay? Now, you need one last type of protein when you get that close. When you're tethered, the next thing you need to do is dock. Okay? And that's basically the vesicle kisses the target membrane. How do you get it to kiss that membrane and actually fuse with it? That takes these types of proteins, okay, and that is the snare proteins. I'm not going to define what snare means because it's upsetting. Just think of it as it's snaring the vesicle so that it fuses directly with the target membrane. And you need two types of snares. You need a vesicle snare, so there's got to be one partner in the vesicle, and then there's got to be a second set, T snares, that means target membrane snares, in the target membrane. Okay, and the general pattern is somehow you've got to have 4 alpha helices that come together to form this V snare, T snare conglomeration. So generally, one of the alpha helices is contributed by the V snare, and actually the T snare contributes generally 3 alpha helices. And what happens is when they get close enough, so the tethers have brought it close enough for the V snare to talk to the T snares, and they form what's called a 4-helix bundle. Where have you heard that term before? It's a motif. What other protein have we talked about that has a 4-helix bundle? Myodine. Myodine. It's a transcription factor. It's a very stable motif. And so what it does is it zippers. In fact, you don't have one set of T snare and V snares. You have a whole family of these around this lower hemisphere of the vesicle that basically stitch it into the target membrane. So it pulls it in and forms a very stable 4-helix bundle, close enough so that basically you're going to squeeze out the water. So that you're going to form this type of structure, and that lipid membrane is going to fuse with that one. And to do that, you need this stable structure because all the water that's right here has to be squeezed out so that the lipids can fuse with the lipids. Right? So let's look at a very practical example of this situation. This is a synaptic vesicle. You know what that is. We've studied it. We were talking about nicotinic receptors and neurotransmission. So this is the nerve cell plasma membrane, like this, okay? And this is a synaptic vesicle loaded with acetylcholine. So what we need to do to cause muscle contraction, so the target membrane is not even shown here, it's down here, okay? We want this synaptic vesicle to fuse with the nerve cell plasma membrane so that the contents inside the synaptic vesicle, acetylcholine, are dumped into the synapse, which is right here. And then it's going to diffuse to the target membrane, which is a skeletal muscle cell, which displays what? A nicotinic receptor, right? Which we know is a non-specific cation channel. Okay, so we're focusing on the nerve cell that causes this release of neurotransmitters. And generally, this is the model for studying neurotransmission. So here are the names. The V snares have very specific names. The V snare is synaptobremin. Okay. The T snare, one of the T snares is syntaxin, which helps you remember that this is at the synapse. Okay. And then there's a second protein that has 2 alpha helices, and it's called the other T snare is called SNAP25. Okay. And so you can see that the T snare, there's 2 classes of T snares, syntaxin and SNAP25. They're going to contribute 3 alpha helices shown in blue, and the V snare is going to contribute the critical 4th alpha helix. This is going to fold into a 4-helix bundle motif, and it's going to pull that vesicle right to the plasma membrane, and they will fuse. Okay? Now, we don't want this to happen just any time, right? Or otherwise we'd all be jerking around all the time. We have to have neurotransmission has to be controlled. Okay, so let's broaden the picture a little bit. What happens is, if in the absence of muscle contraction, there's a protein called synaptotagmin, and I know these names are burdensome, but synaptotagmin is a calcium-dependent protein. And what it does is it gets in here and prevents this 4-helix bundle from forming. If and only if there's calcium present, will it allow that 4-helix bundle to form. Okay, so if there's no calcium, there's no puff of calcium into the cytoplasm. So this is the plasma membrane, this is cytoplasm, this is the synaptic vesicle that's trying to scoot up next to the membrane. Okay, but it cannot fuse because synaptotagmin is disallowing the T snare, V snare complex to form a 4-helix bundle. So this is inhibitory. But what happens is an action potential, that is a wave of depolarization, comes down here and opens a voltage-gated calcium channel, which you know how that operates. It's got a hinge and it opens, and calcium it has to flow in. Why? Because this is 1,000 times or 10,000 times lower in concentration. Calcium is positively charged. This is negatively charged on the inside of the cell. You don't need the Nernst equation to figure out which way it's going. Calcium will flow in here, bind to synaptotagmin, and this thing will swing out of the way, and the 4-helix bundle will form all the way around this lower hemisphere, and this vesicle will fuse. Okay, that will cause skeletal muscle contraction. Okay, now as people get older and they get grumpier and they frown all the time, which uses a lot of skeletal muscles, wrinkles form, right? And so the dermatologist said, maybe we could use some toxin to prevent this from happening constitutively, maybe over 6 months or so. And so they took Botox, which is botulinum B toxin and develop it as a drug that you inject into people's faces to make them expressionless. You can charge them $500 or $600 to do this. So, but what Botox is it is this toxin is a bacterial protease that specifically cleaves the V-SNP. Its target is synaptic vesicle. So if you cleave that, what's going to happen is you can't form the 4-helix bundle, and you prevent the release or the fusion of the synaptic vesicle with the target membrane. So that's how Botox works. The problem is it wears off, and eventually you can regenerate more synaptic brevin, and then you have to go back and spend more money. Okay, that's a lot to digest. So, questions about this? Yeah.
When you were talking about the E115 and the Golgi,
Yes.
would the Golgins also be a Ravifactor, or would it be a different class?
It is. So think of all tethers as being random.
Okay, all tethers are Ravifactors.
Exactly.
And would it be anchored to the cytoskeleton or exo something in the extracellular matrix, or is it just diffusing around freely?
The tethers,
Yeah.
they tend to be single pass membrane proteins.
Okay, but they're not anchored to any component. They're just
They're on the donor membrane and the target membrane.
yeah, like in the membrane. Are they free to move around? Or?
The cytoplasmic domain is whipping around, but it's anchored by a single alpha helix. Yeah. There may be some cases where it's a lipid anchor. That's irrelevant. It's just, it has to be anchored into the membrane somehow such that it displays a long filamentous cytoplasmic domain. That's the rule.
I think she might mean, does it diffuse across the membrane?
No, it does not diffuse. It's anchored.
Like the frap would be,
I'm sorry, the frap.
like let's say if it had a frap, would it diffuse or not?
Oh, I see what you're saying. I don't know, actually, if it's anchored some way inside the donor membrane, but it's sort of irrelevant because it's mobile, and so it needs to be looking for vesicles all over the place. Yeah, actually, I don't know the answer. To what extent is it mobile? But I have a feeling because it's interacting with so many with RAB probably stations it at the domain that's going to fuse with the incoming vesicle. So eventually when they wrap, they need to be stable, so something's going to happen. Okay. Okay, and so the whole point of this is, remember, it's not one T-snare, V-snare, 4-helix bundle. It's a series of 100. Okay, maybe not 100, but it's a lot. They go all the way around the hemisphere of the incoming vesicle, and basically the idea is just to squeeze out the water until these two membranes fuse, and then what happens is whatever proteins were in the vesicle become part of the target membrane. That's the point, and you can tell that by looking at this. Yeah, see? So these membrane proteins are going to become part of the target membrane, including that very stable 4-helix bundle. So now you have a new problem, is how do you unpeel this thing? It doesn't want to dissociate. I mean, it's very happy talking to itself. So what you have to do is you have to employ a special group of proteins to get rid of them so that they can be reused. So you have various accessory proteins. They're called SNAPs. And I'll finally tell you what that means. It means soluble N-ethylamide sensitive factor attachment protein. That's why I didn't want to talk about it. But so these are SNAPs, okay, and it's not the SNAP 25, that's a snare. These are other types of SNAPs that bind to the National Science Foundation, which is N-ethylamide sensitive factor. That's what NSF is. So what this is that's important because it's an ATPase. So it's going to use the power of ATP. It's basically a chaperone that's going to bind to this 4-helix bundle and disassociate it so it can be reused. Okay? So now all these T snares and V snares can be trafficked back to different places and reused without having to throw them away. Okay? So think of NSF as a way to break that apart. It's kind of like an uncoding process, except there's no code. You're just breaking apart the 4-helix bundle. All right. So now we have targeted, we've docked, we've wrapped, we've brought these two together, and so you can see the number of proteins that are interacting cause a vesicle go from a donor membrane to a target is large. And so this is combinatorial biology. You have a few basic components that you recombine it in different ways. It's like our alphabet. We only have 26 letters, but we can create all these billions of words because we recombine them in unique ways. And that's exactly what happens, is the combination of the encoding mechanism, the combination of the cargo recruitment mechanism, the combination of the lipid modification, the RAB, the T-snares and B-snares, the tethers and all that stuff. It all comes together to produce a very precise vesicle tracking pattern. Okay. Ooh, let's take a break here.
Dr. Golgi,
Yeah.
I used the wrong phrase when I said diffuse across the membrane, didn't I? That would imply
You mean
like a...
diffuse in the plane of the
Yes.
membrane? Exactly. And so you don't want to think of this as, it's coming in and it's very directed. I mean, this thing is rotating in a millisecond time frame. And so eventually it's going to whip around wherever it is and link in there. And there's not, the cartoons give a really faulty impression that there's like one or two of them. It's not. It's like 100 all whipping around. But they don't bind each other unless they're specific binding partners. Golgins don't bind to just anything. They bind to P115, whatever it is.
No, it's in our biochemistry class. We have a teacher from science here who works on physiology.
Right.
But he was very distracted, so he didn't go very quickly on what he was covering. But he mentioned that he was working on what I now know called snares.
Snares, okay.
It was interesting.
Wow. So I wonder if he's in microbiology and immunology. You don't remember what department?
Well, I know it has something to do with
the blood.
because we gave a lecture on
Well, I don't know what department that puts him in. He could be in pharmacology. He could be in a lot of stuff. But I'm happy to know that there's somebody that works on snares.
I'll try and get the
Okay.
I'm
Yeah.
just curious if you're going to post the exam 2 feet on one.
Oh, I hadn't even thought of it. Thanks for telling me.
Yeah, sure, yeah.
I completely slipped my mind.
Oh,
I will do that.
Thank you.
Thanks. Yeah.
Okay, so for the 4 helix bundle, or whatever.
Snares.
Yes.
The snare pairs.
Is there only 2 of them, or is it like a big
It's
loop?
a
Okay.
whole stitching that
Okay.
goes all. It's like a stitching machine.
Yeah.
A sewing machine.
All right.
Yeah. Okay, why don't we get started again? I've had a couple of requests to post the Quiz 2 key, which I completely forgot. I'll do that right after this lecture. That'll be posted. One thing I did want to, is a little bit of terminology. If what we've been talking about is where the donor membrane is very different from the target membrane, that's called heterotypic fusion. And that means that the V snare is very different from the T snares. The homotypic fusion means it's like two vesicles fusing together to form a larger compartment, and that occurs a lot. But what's happening is both vesicles, both compartments, have the same set of T snares and V snares. They just interdigitate and use them to fuse to each other instead of a different compartment. So the T snares and V snares on each vesicle would be the same. Okay, so that's a possibility. Okay, what I did want to do is we're supposed to be able to, once we have this knowledge, do something useful with it. And so let's look at an example where it's complicated, and we're not going to learn all the pieces, but you should be able to decipher this a little easier than before we talk about this. This is a very practical example. It's an old friend of ours. This is GLUT4. And so that should immediately suggest to you that that is one of the glucose transporters. And if you remember where you find GLUT4, it's in insulin-sensitive tissues. Okay, so there's two main insulin-sensitive tissues that transports glucose, and that is skeletal muscle, the major consumer of glucose after a meal, and adipose tissue. Okay, so this is a skeletal muscle, and it's waiting to deploy the GLUT4 loaded vesicles to the plasma membrane when blood glucose gets too high. Okay, so you've just eaten a meal, all right? And what happens is, so this is, blood glucose is normal at this point, and so this is the regulated secretory pathway that we're going to talk a bit later. And the characteristic of that pathway is these vesicles are held in check. That is, they're loaded, ready to fuse. Oftentimes they're already tethered, but what happens is, like that synaptotagmin inhibitor, there are a couple of molecules, in this case there's three, synaptomosin and MUNC18C, they're blocking the ability of this V snare to bind to the T snare. That's as simple as it is. In this case, it's not calcium dependent, it's insulin dependent. Okay? And what happens, so here are the players. VAMP2 is the V snare, and that stands for vesicle-associated membrane protein. It's also a unscrupulous woman that uses her sexual advances to entice men. So it's not that type of VAMP. It's vesicle-associated membrane protein. That's on the vesicle that's going to the plasma membrane. And so it can't dock with SNAP23 syntaxin, which are the two T snares, because these guys are in the way. So what happens is blood glucose rises. That tells the beta cell and pancreas to release insulin. And that's a whole process we'll talk about later. And then that goes in the blood, it hits the skeletal muscle cells, and by a lot of fancy jiggery pokery over here, the insulin receptor decodes the message and activates this set of kinases. There's a protein kinase, and a lipid kinase. And so what the sum of this is to phosphorylate this structure so that these inhibitory proteins go away. So it's not calcium, it's a phosphorylation event that removes these inhibitory proteins. That allows the T snares to dock with the V snares. Okay? And so that fuses, and that causes GLUT4 to be delivered to the plasma membrane. There's a 40-fold increase in glucose transport after GLUT4 gets there. And so it sucks glucose out of your bloodstream and keeps your plasma glucose from rising too high. Okay, now, clinically, what's interesting is in people that become obese, and particularly in type 2 diabetes, they become insulin insensitive. Okay, that means that somehow the insulin receptor is not activating this signaling process. And so when blood glucose rises, they can't get the vesicles to fuse to the plasma membrane. Why? So what's the basis for the insulin insensitivity? Well, it turns out that when you have you store lots of lipids in your body, you can actually store them in inappropriate places like skeletal muscle. Should you be storing fat in your skeletal muscle? No, that's pathological. You shouldn't store it in your liver either. That's pathological. What happens is those lipid droplets form in the skeletal muscle, and it recruits SNAP23 to the lipid droplet, removing them from the plasma membrane, making it impossible for this to fuse with the plasma membrane because you've removed the big player that's a T-SNAP component. So that's why you need to lose weight to get rid of those lipid droplets, and then that person becomes sensitive to insulin again, often by just changing diet and exercise, but there are drugs you can take for people that can't do that. Okay. Okay, the next thing we need to talk about is some trafficking. We're still going to the Golgi here. I promise you we're going to get there. So we're going to the Golgi, and we need to talk a little bit about the Golgi itself. And so you know that the traffic going from the Golgi, from the ER to the Golgi involves COP2. Okay, that's obvious. But what you may not understand is once that coating was removed, a lot of those vesicles undergo homotypic fusion and form vesicular tubular clusters. And that's just a fancy term for vesicles forming a tubular larger compartment. And those vesicular tubular clusters fuse with other clusters and become the cis-Golgi network. So one of the reasons it's all bubbly is there are all these vesicles fusing with each other, forming tubules, and there's vesicles fusing with the tubules, and it becomes this bubbly-looking cis-Golgi network, which is the first cisterna, which is sac of the Golgi organelle. Okay. Now, at the same time, what happens is this is not an entirely clean process. Sometimes you have proteins that are ER resident proteins. And what would be a good example of an ER resident protein? It needs to function in the ER. If it goes somewhere else, we must retrieve it. BIP, okay. Anything else? PDI, yeah. Protein disulfide isomerase, and then the peptidyl, prolyl. Those are all soluble things, and they can get swept away and into a vesicle that fuses with the cis-Golgi network. So there must be some retrieval mechanism, and there is. It's a COP1 mediated retrieval process. But that begs the question about how does it selectively pick up BIP or PDI and the peptidyl prolyl isomerase and put it into a vesicle and ship it back. Okay, that's a different story. But we know what kind of coating to use. Retrograde traffic from the cis-Golgi back to the ER is COP1. And we did not cover the details of COP1, but they're codimers, and they have a SAR and various things. They have some sort of GTPase, and we're not going to worry about it. Okay. So here's a vesicular tubular cluster, okay? And so you have homotypic fusion. That just diagrams the fact that you can do that. So if you're BIP, what happens is you have targeting information that is a retrieval signal. Okay? That is near the C terminus of BIP, PDI, and that peptidyl PP isomerase. It's a soluble protein, and it has near the C terminus the KDEL sequence. Okay? And so what that does is it's positive, negative, and two negatives and one positive. So it's a slightly negative charge in leucine. So it's kind of, I always think it's the radio station that's always tuned to the ER. It's KDEL. Okay, and that sequence is recognized by the KDEL receptor. And the KDEL receptor is important because you can't see the coding mechanism required to bring it back to the ER are cytoplasmic proteins. They're COP1 proteins. they cannot see into the ER lumen. So you need a helper to recognize the cargo, and that is the cargo receptor, which is called, oddly enough, the KDEL receptor. So it recognizes that KDEL sequence, binds to it, changes its conformation, and recruits the COP1 coding mechanism, buds this thing off into a vesicle, and returns it to the ER. Okay. Now, that still begs the question of how does the COP1 coding mechanism recognize this cargo receptor? Well, it should work something like the transferrin receptor. So it should be something in the cytoplasmic domain is going to tip off the COP1 coding mechanism to grab onto it. And that's exactly what happens. Okay? So if you're a transmembrane cargo or cargo receptor, you're going to have this sequence. You're not going to have a KDEL sequence, but you're going to display a di-basic or di-lysine motif towards your C-terminus. Okay? And so it's critical that this transmembrane protein be oriented with its N-terminus facing in and its C-terminus facing out, so that this sorting mechanism, it's called a sorting mechanism, to sort it back to the appropriate compartment, is displayed in the cytosol. And that motif is lyse, and then anything, carboxy-terminus. Okay? And so that's poking out here, and that's what tips off the COP1 coding mechanism to grab onto it as a cargo receptor. And then it's attached to the cargo, which is BIP or whatever, and it brings it back to the ER. Now, can you think of a ER resident protein that actually is a transmembrane protein? We talked about at least one last time. Yeah, IR1's a good example. And earlier than that, what's that lectin that is important? Folding, calnexin. Okay, calnexin. So there are cargoes that are actually transmembrane proteins, and they have this motif as well. So you don't have to be a cargo receptor to have that motif. You could be the cargo. And if you're a transmembrane protein, you don't have the KDEL sequence. You have this dibasic amino acid sequence toward your C-terminus. And you get to go back to the ER as well, which is good. Okay.
So
Yeah.
just follow the case number 5 that we talked about.
For which protein?
All of it, KDEL.
Oh, I see. How do you get that to be inluminal? Well, remember there's two ways to get it to be inluminal. You can have a signal peptide on the end, and you have a single transmembrane alpha helix that has no charge information. As long as you have that signal peptide, so that's the insulin receptor example, I think. Okay, and then the second way is if you have no signal peptide, you could just display, where would you display the positive charges so that that's a C-terminus? This is the cytoplasmic side. That transmembrane is right there, so what charge has to be right here? Positives. Yeah. And so that means the C-terminus will display to the cytoplasmic side. So, yeah, those are the two ways to get it, to have that orientation. Good. Okay, and this is just a diagram showing the forward pathway, and you accidentally get these things that you want to retrieve, and it comes back via the COP1 mechanism. And this is a pretty good diagram because it shows the various components of the Golgi, so the cis-Golgi network. You have the medial, so this is cis-Golgi network, cis-Golgi, medial, trans-Golgi network because it's bubbly as well. And that's because things are leaving Grand Central Station. Which is underwater. Okay. Kin recognition. So another way to get things to go together in the right direction is if those proteins selectively aggregate. And that turns out to happen with insulin. So one of the reasons that insulin gets trafficked in the appropriate direction is it coalesces around a couple of different proteins that we'll talk about later. And because they're all sticking together, they get incorporated into the same vesicle. So that's another way. It's called kin selection. So it's a different way of achieving a group of proteins that need to go to the same place all at once. Okay. Now we get to sort of the Golgi itself. And so we have about 10 minutes, and we need to look at, let's remind ourselves of its construction. It's a really bizarre looking set of stacks, but the basic part of this is the cis space is always facing towards the nucleus and the ER, and the trans space is facing towards the plasma membrane. That's a pretty good generalization that you can make about the Golgi. And running through these sacs, these are not stuck together. They're actually on top of the highway that I was speaking about earlier. So there are a series of microtubules that are running from the nuclear area. They're coming out of, some of you might know that, the centrosome. Okay? And they're going towards the plasma membrane. So there's a series of highways going this way, and these stacks are actually attached to those microtubules by motor proteins. So this thing is moving along. And that's actually explains one of the ways that we have to go to the next PowerPoint. That explains how you get these trafficking incidents to move stuff through the Golgi stacks. There's a couple of choices of how you might do that. The stacks themselves are very specific about the type of proteins that we have. Remember, one of the big things that the Golgi does is that it modifies the sugar residues. So glycosylation starts in the ER, but it finishes in the Golgi. So there's all these enzymes, and you do not have to memorize these. Okay, we will only talk about one specific one that has to do with lysosomal signaling. So we're not going to remember, but the point of this diagram is to understand there are particular enzymes that have to go in each one of these stacks. So whatever mechanism you have of moving traffic through the Golgi must account for the fact that each stack has its own enzymes. So this is moving, but yet it's stationary. So it doesn't make sense, right? It's difficult. Okay? So somehow all these components have to be in the right place, and then this place has to sort all the traffic to go to these three different destinations: lysosome, plasma membrane, and secretory vesicle. And there are a couple of explanations for that. The first is the vesicular transport model, and that is each cisterna is stationary. It's somehow linked to something, some matrix that holds it in place, and what's actually moving is the vesicles go between the stacks carrying the cargo. Okay, so if you want to send stuff forward, you go this way, and so this vesicle blebs off here, and it's called a transport vesicle if there's no other purpose for it. Okay? So if it's just going between two stacks and you don't know what else to call it, it's called the generic term is a transport vesicle. And so it just goes forward, and it goes to the next guy, and this accounts for the fact that each stack has its own enzymes because they don't move. Okay. Now, so this is the vesicular transport model, and there's a second model that's called the cisternal maturation model. And most people actually believe this one, and there's more evidence for it. And basically what this is what's happening is this vesicular tubular clusters forms the cis-Golgi network, which then binds to these motor proteins that attach it to the highway, which are microtubules, and the cisternae themselves are moving along. Okay? Well, what happens is after this cis-Golgi network moves forward, it's replaced by another group of vesicles that are coming from the ER. So it's continually in flux. So every cisterna is moving forward. So then you have to go, well, how do you keep each cisterna to have the same complement of enzymes? Well, you keep sending the enzymes backwards, retrograde, using COP1 mechanism. Okay? And so if this moves forward, you send all the enzymes necessary for this guy backwards. So it always ends up in the cis-Golgi network. If you're a medial Golgi and you move forward, you send your cargoes backwards to keep this always having the same enzyme. So it's the sacs that are moving, and the vesicles are keeping each cisterna characteristically with its own set of enzymes. So it's really I like this model for a couple of reasons. One is that it actually involves both things. It's vesicular trafficking, and it agrees with the fact that we see under the microscope, this thing moves, okay? The other thing is, if you're going to secrete something big like collagen, it's a large protein, it is not going to fit in a vesicle. So you can't have this model. This model does not explain trafficking of collagen, because what you have to do is package collagen into this vesicle to move it forward, and it won't fit. So what you can do, though, is if you put collagen into this cis-Golgi network, it can pack in here and it can move forward as a large package to be carried forward and then exit into the plasma membrane. Okay, so this one tends to be the one that people like the best, although there's still some controversy. Okay. All right, I think we'll stop there and pick it up next time. And I hope I see some of you tomorrow at noon over at the Health Sciences Center. I probably just need to sit down and play with it. I think I'll put it on. Yeah, put it on. Yeah, that's
Hey,
fine.
how are you? I'm all right.
Can recognition be observed in retrograde movement too? So if, like Vin was moving that.
It could.
And then